like to welcome all of you to this really exciting series that, in fact, was the brainchild of the Brown Chair holders and our university scholars. The idea of engaging our faculty that do amazing scholarship with the community. And so what you're going to hear are two brief lectures about original scholarship and original work by some of our faculty here. And then we're going to follow it up with something very unusual, a chance for you to respond. And I want to make sure you understand you have a job here. So you are a working audience. We're not going to have a Q&A like you would normally have. Instead, your mission is to create these Paideia moments, meaning as you listen to these two speakers, discover connections between what they are saying and what they are doing. And you will be given an opportunity after we hear from both of these scholars for you to share what you've heard. If you have questions for our speakers, they'll be glad to entertain them during the reception. But during that last third of our time together, it's going to be you sharing just what you hear, and I'll try to facilitate that. And I'm not afraid of cold calling. So if you don't volunteer, I'll just pick someone in the audience, and so you're all on call. With that, I'd like to introduce my colleague, Laura Hobgood-Oster, from the Religion Department, who will actually officially kick off this event. Thank you so much for being here, everybody. Thank you.
Thank you, President Berger. And I also want to thank the staff and faculty in the Fine Arts Building, and also for Paul Sicard for helping get all of this together today and for making this space available. As Dr. Berger mentioned, there's a theater production class going on back here now, and they promised to be as quiet as they could. But you might hear a few bangs from going on behind the curtain there. So now it's an honor to introduce the first of our two inaugural Paideia Connections scholars, Dr. Maria Todd. Dr. Todd joined the faculty of Southwestern University in 2001 and is currently Associate Professor of Biology. She received her Bachelor of Science in Biology, focusing on molecular genetics from the University of Sussex in England, so you get to hear her wonderful, beautiful British accent. Cambridge University was the next stop in her academic life, where she earned a PhD in biology in 1990. For the next 10 years, she worked at the University of Texas and the University of Colorado on the Human Genome Mapping Project, studying a lung cancer tumor suppressor gene. That's why we're introducing people in totally different areas. That alone, lung cancer tumor suppressor gene, was sort of a tough one to get. I read it several times. Obviously, very, very important work that she has done for years. Then she also worked on cell cycle gene defects responsible for the early stages of breast and ovarian cancers. Since her arrival at Southwestern University, Dr. Todd has worked closely with two colleagues in biology, Dr. Cuevas and Dr. Scheller, on the metastasis of breast cancer. Her talk today builds on that work, breaking the ties that bind, the role of tight juncture deregulation in breast cancer metastasis. Please join me in welcoming Dr. Todd.
Thank you, Laura, for that very kind introduction. And thank you, President Berger, and the Brown Chairs for inviting me to participate in today's Paideia Connections event. I'm really delighted to have this opportunity to present alongside my dear friend and colleague, Dr. Alison Kafer, and to have this rather rare opportunity to discuss with such a diverse audience some of our latest breast cancer research. Most of all, though, I'm looking forward to what you have to say in about 45 minutes' time when Alison and I have done our bit. So there are, I think, few experiences in life that generate in us as much abject fear and dread as a diagnosis of cancer. This despite the declining cancer mortality rates over the last 10 to 15 years. But let's face it, a diagnosis of cancer elicits a visceral rather than a rational response and has little to do with health statistics and more, I think, to do with the nature of the disease itself. For cancer is a disease that arises from our own once completely healthy cells through the accumulation of damage to our genetic material or our DNA, usually over several decades. The resulting transformed cells bear very little resemblance to their normal cell counterparts as they proliferate in a rampant, uncontrolled manner. Benign tumors are localized and usually non-life-threatening unless they happen to develop and interfere with the function of a vital organism, or organ rather. In contrast, metastatic or malignant cancers, those cancers that have broken their connections with the primary tumour and moved to another organ in the body, are responsible for upwards of 90% of all cancer-related deaths. And it's the metastasis of cancer that has occupied my collaborators, Dr. Maria Cuevas and Dr. Rebecca Scheller and I, for the last several years. And that's what I'm going to talk to you about today. What are some of the causes of cancer metastasis? And in particular, we're interested in breast cancer metastasis. Why are we interested in metastasis of the breast? Well, as you know, the rates of breast cancer continue to rise worldwide, but particularly so in the Western world. And here in the United States, about 1 in 8 women will expect to receive a diagnosis of breast cancer during her lifetime. Now, while it's relatively straightforward to cure early stage breast cancer, it's far more of a therapeutic challenge to effectively treat malignant or metastatic breast cancer. What I want to do now is show you the general stages of breast cancer development. These rectangular boxes represent epithelial cells. Epithelial cells are cells that cover organs and line tubes and vessels in our body, and 80% of all cancers arise from this particular type of cell. There are a couple of things that you need to know about epithelial cells. The first thing is that they divide and proliferate in a strictly regulated manner. A cell will divide until it's surrounded on all sides by other cells, and then they send signals to one another saying, stop dividing. And because of this, epithelial cells grow in these beautiful single or monolayers in a highly structured way. The other thing I want you to know about epithelial cells is that they cannot move. They are anchored to one another by very tight connections, and they're anchored to this underlying cushion of proteins and sugars that we call the basement membrane. Let me give you an example of how they're anchored. Would you, if you are able and willing, raise your hand if you have a mole anywhere on your body? Don't tell me where it is. I've lived a sheltered life. Okay, so lots of you have moles. Lovely. Would you raise your hand if you have ever woken up one morning and found that your mole has moved to another part of your body? Thank goodness for that, or I would be calling the ambulance. And that's precisely because epithelial cells in moles are anchored to one another. They can't take busman's holidays and move somewhere else in the body, okay? Now, oops, let me see. Okay, sometimes genetic mutations or damage to our DNA occurs, and in normal healthy cells, that damage, shown here by the thunderbolt, is dealt with immediately. The mutations are fixed. If the damage is extensive, the cell will actually commit suicide for the sake of the host organism. Sometimes, however, this damage is not repaired quickly. And what happens is that the cell accumulates further genetic damage, and we start to see changes in the appearance and structure and function of our cells. They don't necessarily turn orange. That's just to show you that the cells are changing. Over time, further accumulation of mutation leads to the build-up of cells. They start to pile up on top of one another instead of forming these lovely single monolayers that I told you about earlier. As the mass increases in size, so does its hunger for nutrients. So cancer cells produce chemicals that allow the formation of new blood vessels that now feed the tumor, and we call this angiogenesis. Again, over the course of years, more genetic mutations accumulate such that some cells are able to break their connections with their neighboring cells and actually start to move away from that primary mass break their way through the basement membrane and enter the blood or lymphatic system. And in the case of breast cancer, those cells metastasize to 3 major places: the lung, the bone, or the brain. Okay? Now, metastasis is an active, dynamic process. So when we study metastasis, we don't want to study frozen lumps of tumor, we want to study living cells. And I'd like to show you some of our beautiful cells. And they are beautiful, don't you think? Here on the left-hand panel are normal breast epithelial cells. This stands for human mammary epithelial cells. We obtain these cells from women with healthy breasts undergoing breast reduction surgery. These cells are mortal, which means they have a finite lifespan. In practice, that means we've got a month to work with them before they pop their clogs. We have to work very quickly with these cells. I want to point out something. These beautiful blue crystalline-looking structures are the nuclei of the cells. That's where the genetic material is housed, okay? And another point to make about these cells is that it's actually very difficult to see where one cell ends and another cell begins. And that's because those connections between the cells are so very tight. Compare the appearance of our normal breast cells to breast cancer cells. Now, of course, they've got very cumbersome names to protect the identity of the women who donated the cells. But both of these cell populations were obtained from women who had breast cancer that had metastasized to the lung. So these cells were removed from the lungs of 2 women. The MCF7 cells, as you can see, are very clearly demarcated, which implies that the connections between them are not quite as strong as between normal healthy cells. You can also see that the MCF7 cells are starting to pile up on top of one another a little bit. They're not as organized. Now, the NDA MB415 cells barely make any connections at all. Okay? They're very independent. They're not signaling one another or talking to one another. Something that's important to know about these cells is that the women who donated them both died in 1970. But their cells are alive and kicking in our lab in Fondren-Jones right now. These cells are immortal. They'll outlive every one of us and our children and grandchildren as long as we keep feeding them. What conditions favor metastasis? How is it possible? Well, first of all, cancer cells need to break their connections with other cells within the primary tumor. And once they've broken these connections, they have to physically move. And I've told you that they can't move. So they have to manage to move if they're going to metastasize so that they can make it to a blood vessel. Once they're in a blood vessel, these cancer cells have to survive these extremely dynamic forces in a fluid environment in the absence of anchorage to any other cells. It shouldn't surprise you to learn that only 1 in 10,000 cells actually survive when they move away from the primary tumor. But don't feel sorry for them. So today I'm going to tell you about a couple of experiments that address these first 2 conditions. I'm not going to tell you about experiments that address the 3rd condition for 2 reasons. Time is limiting, and I haven't done the experiment yet. So come and see me this time in a year, and hopefully I'll have some data for you. So let's look at this first condition. Cancer cells need to break their primary tumor connections. Well, how do they do that? Well, cells are held together by these components called tight junctions. So in order to break their connections with one another, they have to break their tight junctions. But what is a tight junction? Well, here on the right-hand side, you see 2 adjacent cells. This is one cell, and this is another cell, and the edges or the membranes of the cells are held together by tight junctions. And they are so tight that fluid cannot pass between these 2 cells. If you've ever woken up at 3 o'clock in the morning and wondered why the contents of your stomach or bladder don't leak out, it's all because of tight junctions. Now you know the answer. On the left-hand panel, you're seeing the molecular structure of these tight junctions. There are many proteins involved in them, but the major family of proteins are called the Claudin proteins. And the Claudin proteins join themselves and their cells together like Lego bricks. It's a really tight connection, okay? Now, at the molecular level, the amount of protein produced is really important. If you have 1.5 or 2 times too much of a protein, that's enough to destabilize these tight junction connections. So it's very important that the levels of these Claudin proteins are carefully regulated. So we thought that it would be an interesting idea to assess tight junction destabilization in metastatic breast cancer cells. Well, how do you do that? Well, the way you do that is to actually measure and compare the amount of different Claudins produced by normal cells versus metastatic cells. And any deviation from the norm would indicate that this plays a role in metastasis. Well, here's the experimental design. What we basically do is we grow up a culture of normal breast cells and we extract the protein from those cells. And then we grow up cultures of our 2 metastatic breast cancer cell lines and we extract proteins from those cells. And then we use Claudin antibodies that are specific for certain types of Claudin proteins to measure the levels of the Claudins within the cell. There are 24 different Claudin proteins, but I'm just going to show you data from 2, because I'm kind that way. Okay, so this is my most favorite data in my entire 25-year career. So, just so that you can be suitably impressed, I'm priming you. So this band here in pale gray shows us the total amount of Claudin-3 protein produced by normal breast epithelial cells. Your breast epithelial cells are producing this much Claudin, okay? Now compare that with the whopping amount of Claudin-3 protein produced by the metastatic breast cells. Whopping is a technical term which means a great deal of, okay? This is actually more than 200 times more than normal breast epithelial cells produce. Easily enough to destabilize tight junctions, as you can imagine. Well, we weren't content with Claudin-3, so we also looked at the level of Claudin-4, and we're equally impressed. Claudin-4 is produced at barely detectable levels in normal cells, but look how much we're seeing in the metastatic breast cancer cells. Okay? Not quite as much as Claudin-3, but still very impressive. So this data is very good evidence that we're seeing destabilization of tight junctions in metastatic breast cancer cells. We have other data to support this, but I don't have time to show that today. Well, just in case you're not as easily impressed as I am by blobs, I'd like to show you what Claudin-3 levels look like in cells. And so here we've taken exactly the same Claudin-3 antibody, but we've tagged it with a green fluorescent protein. So everywhere you see green, you're seeing Claudin-3. And I'm just showing you the data for MCF7 cells. Aren't they pretty? So these are the cells, and you can see the Claudin is localized in the edges or the membranes of the cells, exactly where we'd expect it to be. But we also find evidence of Claudin-3 inside the cells. It has no business being inside the cells. It's up to no good. And that's another line of investigation that we're currently following. Is Claudin-3 taking on these kind of illegitimate responsibilities in other, or functions in other parts of the cell? Well, because the Claudin-3 data, particularly in MCF7, was so dramatic, the second experiment I want to show you focuses just on this pairing. So we've talked about the first condition that favors metastasis. The second condition is that cells have to detach from the primary tumor and move, physically move. And we wondered if these elevated levels of Claudin-3 protein might actually enhance motility or movement of the cells. So how do we assess the effect of these high levels of Claudin production on cellular motility? Well, we compare the motility rate of breast cancer cells producing abnormally high versus normal levels of Claudin-3. And we say, how fast do the cells move when they're producing high levels of Claudin-3? And can we slow them down if we bring those levels to normal levels? Well, how do you do that? How do you shut down high level of Claudin-3 to normal levels? Well, we use a technique that involves something called small interference RNA that can target the production of Claudin-3 and make sure it's present at a very low level. And I've just written in 20 words something that took me 20 weeks to optimize, just so that you know. So here's the assay procedure. We take these tiny little wells that are like very small Petri dishes, and into those we place a rectangular insert, shown here in orange. And then we take MCF7 metastatic cells producing really high levels of Claudin-3, and we plate those around the insert, okay? And then we do the same thing with MCF7 cells producing normal levels of Claudin-3, okay? Once the cells have grown to confluence, once they've covered the well, we remove the insert and we actually measure the rate at which the cells move into the space that we've created. And here's the data. Let me first draw your attention to this little figure down here. This shows you untreated, or MCF7 cells that are completely untreated, and this is the amount of Claudin-3 that they're producing. And these are the same cells that we've treated with small interference RNA to bring the level of Claudin-3 down. If we look at this top row to begin with, these are MCF7 cells producing the high levels of Claudin-3. And here we've just removed the rectangular insert, and then over a 3-day period, we monitor the rate at which those cells fill in the gap. And you can see they're moving quickly. So high levels of Claudin-3, they're dashing and they're filling in this gap within a 3-day period. But when we shut down Claudin-3 to normal levels, and we do the same experiment, you'll see that even over a 3-day period, they're not able to move into that insert. So these data indicate that high levels of Claudin-3 actually enhance cellular motility. And importantly, when we interfere with the production of Claudin-3, we can slow down motility. And so to summarize, we've shown that Claudin proteins are produced at very high levels in metastatic breast cancer cells, and this helps to destabilize cell-to-cell connection and increase the rate of movement of breast cancer cells. So we are among a very small group of scientists worldwide working on this, but we feel that we've shown that destabilization of tight junctions appears to play a role in the metastasis of breast cancer. So the most important part, of course, is how do we translate basic research findings into clinical applications? Well, currently, the way that we diagnose breast cancer is to palpate a mass, which means you feel a lump with your fingers, or your physician feels a lump for you, or we image a mass using mammography or digital ultrasound or MRI. But in order to feel a mass or image a mass, the tumor's already quite advanced. So we need blood markers for breast cancer in the way that we have blood markers for prostate and ovarian cancer. And we're quite hopeful that elevated levels of Claudin-3 may act as a good diagnostic and possibly prognostic tool for cancer, for breast cancer. But I'm most excited about the potential therapeutic applications of our research. We've shown in the laboratory that we can shut down the level of Claudin-3 and decrease cellular motility or movement. We want to be able to do that in women, okay? So how do you decrease Claudin-3 levels in women, in their tumors? Well, you can use siRNA. How do you get the siRNA to the tumors? Well, you can use nanoparticles. And there are two groups at Texas Methodist Research Institute and Rice who are doing just this at the moment. They're using nanoparticles to deliver siRNA to tumors. So I'm very hopeful that in the next decade or two, we might see some clinical application of our research. And so I'd like to end as I began, with on a sincere note of gratitude to my wonderful collaborators, Dr. Maria Todd and Dr. Rebecca Scheller. I love you, and you're a constant source of inspiration and joy in my life. And talking of inspiration and joy, I've had 3 amazing students who've worked on this project with me throughout the years. Britt Piana, now a practicing OBGYN, Bronwyn Tyler, who's about to go to medical school, and Heather Petty, who is about to embark on a master's in translational medicine. Dr. Jonathan King is our wonderful collaborator at Trinity University, and we need a lot of money. This work is really expensive. So we spend a lot of our time applying for external and internal grants, so thanks to everyone who's given us money, and I'd like to end and thank you for listening.
Thank you, Professor Todd, for that presentation, which was as informative to this musicologist as it could possibly have been. Exciting, very promising work. I'm just delighted. I'm Dr. Michael Cooper. I'm the holder of the Margaret Root Brown Chair in Fine Arts, and I'm one of the organizers of this series, and I'm here to briefly introduce our next speaker, who, frankly, really needs no introduction, but I must do it anyway. So, our next speaker is Dr. Alison Kafer, who received her Bachelor of Arts degree at Wake Forest University in 1993, before going on to receive the MA and ultimately the PhD from the Claremont Graduate University in 2005. Over the course of her career, Professor Kafer has not only established a strong record of grant support for her research, but also distinguished herself as a teacher, receiving Southwestern's Exemplary Teaching Award in 2008. Quite an honor that. Professor Kafer has also firmly established herself as a leading voice in the rapidly growing and increasingly important fields of disability studies and feminist and queer theory. She has authored chapters and books published by Duke University Press, Gallaudet University Press, Indiana University Press, Rutgers University Press, and Routledge, among others. I'm sure I missed some in there. And articles in journals including Biography, an Interdisciplinary Quarterly, Sex Roles, and most recently, the Journal of Women's Studies. She recently guest edited a special issue of Disability Studies Quarterly, which is due out in April, and is currently working on an essay on trauma and disclosure, as well as a new book on skin. We'll have to wait to see those products, of course, but in the meantime, there's still plenty of time to read her latest book, which is titled Feminist, Queer, Crip, and was released by Indiana University Press in 2013. Please join with me in welcoming this outstanding scholar, teacher, and colleague to the inaugural episode of Paideia Connections, Professor Alison Kafer.
Thank you very much. That was too kind of an introduction, Michael. Thank you very much. Before I go any further, I just want to thank all the faculty and staff who did so much work to make this afternoon happen. There was much organizational detail, for which I am very grateful. And I also would like to thank President Berger and the Brown Chairs for inviting me to be here. It's a great honor to get to talk about my work with all of you, particularly because so much of the work that I have done has been informed by my students and my colleagues in feminist studies. So thanks to all of you. I want to give you a little overview of what I'm going to do during my time. I'm going to talk about my recent book called Feminist, Queer, Crip. So I'm going to give you a summary of the book. I'm also going to talk about the terms that I use and why I use them. And then I'm going to take you through a few examples. So the title of my talk today is called Edgework, Disability and Relation. The title for this talk comes from Audre Lorde, who's a Black lesbian feminist poet and theorist. And although I didn't use this title for the title of my book, her work has really informed my work as a whole. So it seemed like a great way to introduce the topic to you this afternoon. So this is a quote from Lorde. "We have chosen each other and the edge of each other's battles. We seek beyond history for a new and more possible meaning." So I read this quote and I think about what it means to choose each other or to choose each other's battles. So I think about questions of coalition, of alliance, of doing cross-movement work. But I also think about these things through the lens or rubric of choice, of desire. What does it mean to choose each other and to choose each other's battles. But thinking also about edges. She says we're choosing the edges of each other's battles. So this phrasing suggests to me that our battles don't line up perfectly. They don't map perfectly onto each other. So for me, this raises questions as well about struggle, disagreement, debate, contention, how doing cross-movement work is going to require these kind of sort of contentious relationship. And I'm also really intrigued by her phrasing about new and more possible meetings. What does that mean? What does it mean to be working toward new and more possible meetings? So it makes me think about visions of the future. What is the future we are all working toward? So I'm a feminist studies professor. I teach courses, obviously, in feminist studies, but also in environmental studies here and race and ethnicity studies here. Queer theory is a topic that surfaces in much of my work, and my research is really centered in disability studies. So for me, sort of sitting at the intersections of these different fields and thinking about Lord's work, I find myself thinking about disability and relationship to other social movements, and thinking about the role of disability in ideas about the future, ideas about new and more possible meetings. So when I think about stories about the future and the role of disability or disabled bodies in those stories, what I notice is that disability often surfaces as the future that nobody wants. So that a good future is one without disability and disabled people, and a bad future is one with disability and disabled people. So that disability and disabled bodies serve as the best illustration of what bad futures look like. So let me give you a couple examples. Think of novels that you've read or films that you've watched, and how often you can recognize the villain by the fact that they are disabled, right? It's almost always a guy, and he's missing a leg, or he's in a big, horrible wheelchair, or he's got this weird prosthetic, or he's wearing a mask over one of his eyes, right? Disability is what serves visually as a sign that this is a bad future. This is a direction we don't want to go down. So that's interesting, but I'm actually even more interested not so much in literature and film, but in sort of the stories we tell ourselves about the future. So looking at more daily interactions. So let me give you a couple of different examples. It is not at all uncommon for strangers, people I don't know, to come up to me in public and say, if I were you, I would kill myself. There is absolutely no way that I could live in a wheelchair in the way that you do, right? So that comment, again, suggests this assumption that a life with disability and a future with disability is a future that we all agree we don't want, right? Or to take a different example, in some parts of the country, in some parts of the US, 90% of women, of pregnant women who receive diagnoses for Down syndrome choose to terminate those pregnancies on the basis of that diagnosis. So again, there's this assumption that disability is this future that we don't want, such that some women who actually go ahead with carrying those pregnancies to term, and so they're out in public with their children with Down syndrome, report having strangers coming up to them and chastising them for not getting prenatal testing, right? How dare you bring this child into the world, because we all know that this child is going to have nothing but a bad future, right? A bad life. And obviously you can sort of see how this assumption is so taken for granted that strangers find nothing at all bizarre about going up to people they don't know and saying, if I were you, I would kill myself, or your child has no right to be here, right? There's this assumption that we will all agree to this idea. What's interesting to me, though, is that this assumption is found not only in sort of mainstream interactions, but also in feminist and queer theory. So as a scholar, when I want to unpack this assumption, I go to feminist studies, I go to queer theory to find possibilities of thinking about futures differently, but I find this exact same assumption, the same notion that disability or disabled bodies are the best way to illustrate the futures that none of us want. So where does that leave somebody like me who wants to articulate sort of a queer, feminist, crip politics? So before I go into examples of alternatives, so ways to think this differently, I want to talk a little bit about the words that I use and why I use them. My understanding of disability is very much related to my location or my placement in feminist studies. So the work that I do within feminism and as a feminist. Feminist theory is actually what led me to disability studies. So just that as I had seen feminist theorists sort of question the naturalness of femininity or to challenge essentialist notions of the female body, I could do the same thing about disability or disabled bodies. So just as feminists sort of question this notion that we all know what women are or what women do, I could use theory to also sort of question how we assume we all know what disability is or what disability does. So feminist theory is what gave me the tools to think about disability and our assumptions about disability and disabled bodies, and how those assumptions lead to things like resource inequalities or social discrimination. So it was through reading feminist theory that I came to understand disability as a political category rather than simply personal tragedy or individual pathology. And it's that shift, thinking about disability as a political category. So it's a category that has a political history and a cultural history, as a category that has material ramifications on people's lives, that makes it possible, I think, to begin thinking about disability or futures of disability differently. In other words, once we see disability as something that we can debate, as something that we can discuss, then it's possible to imagine it as something else. So now let me talk about queer and crip. Now, crip, as you might imagine, as you might be able to see by looking at it, is indeed an abbreviation of the word crippled. So both of these words, both queer and crip, have histories as epithets, but they have been reclaimed by many theorists and activists as signs of radical politics or of a politicized community identity. They both often can serve as umbrella identity terms. So queer can refer to lesbian, gay, bisexual, transgender, genderqueer, intersex, and so on. In the same way that crip doesn't necessarily only refer to people who are crippled, who have some sort of mobility impairment, but can also refer to people who have sensory impairments or madness, psychiatric impairment, cognitive impairment. In other words, anybody who has any kind of disability. Now, these are still very contested and controversial terms. Even people who use these words, queer and crip, disagree as to their meaning or their usefulness. One of the reasons that I use them is because I'm really intrigued by their promise for coalition politics. So I'm intrigued by this notion that they can serve as umbrella terms representing a lot of different positions. But another reason I use them is precisely because they are contested. I like the fact that not everybody agrees about what they mean, because I think that means there's a room to struggle, to sort of figure out what the edges of each other's battles are. And I like the fact that they make people nervous, that they suggest sort of an urge to shake things up, to change people, to make people think about what their everyday understandings are of bodies and of minds, or of normalcy and deviance. And I also think it's important to think about these two words in relationship to each other, because queerness continues to be thought through the lens of disability, with both queers and crips rendered unnatural, sick, degenerate, deviant. So these are words that can help us forge a politics. In other words, I am interested in these words not only as sort of identities, but also as political affiliations. And so when I think about political affiliations, it brings me back to the Lord quote at the beginning about new and more possible meetings and the possibility of imagining disability futures differently. So informed by feminist and queer theory, informed by critical race theory and ethnic studies, is it possible to imagine crip futures? For me, one way of doing that is to look at the work of theorists and activists who are engaged in cross-movement work. So theorists and activists who are refusing to accept this notion that disability and disabled people constitute the future that no one wants. And I think what this means and what it requires is looking at disability in relation, or crip relation. So thinking about disability in relation to other movements for social justice, racial justice, economic justice, environmental justice, reproductive justice. So what I want to do for the rest of my time is take you through a few of these examples. So potential moments of coalition, potential moments of possible interaction. And I've chosen the sites that I'm going to talk about today precisely because they are difficult, because they're sites where the edges of different battles bump up against each other in ways that are unpredictable and often uneasy. So the first of these that I want to talk about is one I hinted at earlier, which is relationships between disability movements and reproductive health, rights, and justice movements. Now, disability rights movements and reproductive rights movements very rarely talk to each other or engage with each other. In fact, they often actually see the other movement in an oppositional or hostile way. And this divide is understandable. So reproductive rights advocates often use disability as a justification for abortion, so that disability serves as the ultimate reason as to why abortion should be kept safe and legal. There's not much room for a disability rights movement position within that framework. In other words, it's really hard to find common ground with organizations that take for granted the undesirability of disability. Particularly, I think, given the statistic I mentioned earlier about the frequency of termination around diagnoses of Down syndrome. However, because abortion is so consistently under attack in this country, reproductive rights advocates understandably are wary of disability critiques of prenatal testing or abortion practices because they hear them as threats to abortion. And indeed, one of the new fronts of anti-choice or anti-abortion movements is to make illegal abortion on the basis of disability diagnosis. Several states have made this move, and it's an attempt to make sure that disability rights movements and reproductive movements continue to see themselves as opposing. So reproductive rights movements are understandably wary about any kind of raising of issues of disability or questions of prenatal testing. So what I talk about in my project is that perhaps reproductive justice approaches, so reproductive justice as opposed to reproductive rights, might be a more useful framework thinking about disability and reproductive issues together. Reproductive justice frameworks are pioneered by women of color, and what the folks in these movements have done is say that abortion cannot be the only issue, that advocates and activists for reproductive rights and health need to talk as much about the right to parent and having support in choosing to carry pregnancy to term and the resources that one needs as much as the right to abortion. So let me say some of this differently. My concern is that when we force women and reproductive movements living in an ableist culture, so a culture that presumes disability is a life or a future no one wants, when we force women in an ableist culture to prove that their abortions are justifiable, disability is always going to be an effective justification for preserving, at the very least, a minimal right to abortion. So in other words, when the legality of abortion hinges on some pregnancies being seen as abortable and obviously abortable, then drawing lines between impairments becomes inevitable, so that we start having cultural conversations about whether or not it's okay to abort for blindness, but not for deafness. It's okay to abort for Down syndrome, but not for a malformed hand or foot. This condition is too severe, and that condition is not. There is no way for disability movements to win in that kind of conversation. So making disability do the work of defending abortion may be effective in the short term, but it does so by trafficking in discriminatory stereotypes about disability. And as we are seeing now, its long-term effectiveness is doubtful because it opens the door, it makes permissible the practice of asking and expecting women to justify their decisions for abortion. So what I'm arguing is that both movements can benefit from taking each other into account, so that rather than relying on the specter of disability or the specter of disabled children to motivate public response or public support for abortion, activists instead should be supporting all reproductive choices, taking a reproductive justice framework, including not only the right to abortion for any reason, but also the right to parent and the resources one needs to do so. So let me take a different example. Environmental justice movements and their relationship to disability. Disability tends to surface in environmental discourses only as that which should be avoided. So disability surfaces in this rhetoric or in this framework as a tragic mistake caused by unnatural incursions into or disruptions of the natural body or the natural environment. So my question is this. Is it possible to build environmental justice movements or anti-toxics movements that don't rely on the specter of disability to motivate public response? Part of my argument is that that kind of deployment of disability is ineffective, that focusing exclusively on disabled people or often disabled fetuses as the signs of environmental injustice erases the way that all of us are affected by toxic pollution and contamination, not just those of us with visible or apparent, quote unquote, abnormalities. So the disability studies scholars and activists can join with environmentalists to ensure that descriptions of the possible impairments linked to toxic exposure don't replicate ableist language and assumptions. So let me show you an example. This is a passage, a quote from the Collaborative on Health and the Environment. Developmental delays, learning disabilities, ADHD, and behavioral disorders extract a terrible toll from children, families, and societies. The cost associated with caring for these children can be high for families and society. Special education programs and psychological and medical services drain resources. There's a long history in this culture of talking about the economic burden of disabled people, and particularly of disabled children, and this quote certainly builds on that history. Well, what's more disturbing and more interesting to me is the phrasing of the quote itself, because what it does is it suggests that disabled children and the services that disabled children need are the ones responsible for draining public resources, as opposed to the polluters who created the poisons in the first place. So if we start thinking and looking at environmental justice through a disability studies lens, we can avoid this kind of misdirection and place the blame where it belongs on the polluters as opposed to the disabled children. Another way to think about disability and environmental relationships is to think about, are there ways that we can use disability laws to combat the poisoning of neighborhoods or combat pollution of this sort? So for example, could we use federal and state disability laws to reduce environmental hazards for entire communities? So if we have children in a public school who have respiratory illnesses that would be negatively impacted by the use of chemical pesticides in those schools, can we argue that the use of pesticides in those schools therefore makes the school inaccessible to those children with respiratory disabilities, and therefore those pesticides shouldn't be used, which as a result would benefit all the children in the school and not just the children with disabilities? There's a group of activists right now in Oakland who are trying to protest the widespread application of chemical herbicides in public parks by arguing that the use of those herbicides in the parks will make those parks inaccessible to people with disabilities. And so they're wanting to stop the city from using those pesticides by saying that it's going to make an area inaccessible. So again, it's a way to think about disability laws or disability rights to benefit all people and not just those people, quote unquote, with disabilities. I talk about more examples in my book, and I'm not going to talk about them in much detail right now, but I just kind of want to mention them to get you thinking about them. So could we think about a broad anti-institutionalization movement as well? So could people who are doing prison abolition work together with disabled activists who are working to protest the warehousing of disabled people in nursing homes or state institutions, so that we can really talk about anti-institutionalization in a much broader framework? Or to take a different example, can we see public bathrooms or public restrooms? And I love talking about public restrooms, so please stop me afterwards to talk about them. Can we see public restrooms as sites for coalition around people who find their use of public spaces restricted by the lack of access to restrooms? So this would affect, obviously, disabled people who are encountering public spaces that don't have accessible restrooms, but it might also be a way to make coalitions with homeless people who are often excluded from public restrooms, but also people who find their use of public restrooms restricted on the basis of gender identity or gender presentation. So can public restrooms be a place to get all of us to think about access and activism around access in a much more broad way than simply thinking about quote unquote disability? I close the book and I'm closing this talk with these examples because I think they're such fruitful, generative places of thinking about coalition and thinking about how we can make connections across movements. And I also like them because I think they can show examples of how disability studies or a feminist queer disability studies can help us see old problems differently. It can raise new issues, it can offer new perspectives on the issue. And to return to the Audre Lorde quote at the beginning, I think it's a way to think about edges, right? The edges of each other's battles as sites of connection and as fruitful sites of connection. So I think at that point, I'm gonna stop so we can make room for your connections between things. So thank you.
Weren't those marvelous? Just spectacular. Well, I've got a bunch of notes here that I'm delighted to share with you, but I'd rather start with individuals that maybe want to share with us. I don't, want long soliloquies here, but just one connection between these two marvelous presentations. Yes, please, this woman in the purple, and then we'll come down to Helene.
I found it interesting that the tight junction that Dr. Todd spoke of as being necessary can be analogous to the tight junction that Dr. Kafer is suggesting between groups, without which you get cancer metastasizing, without which you have groups going separate ways and maybe doing more harm than good.
Thank you. Helene. Right here, Ben.
Both of your talks taken together suggest that we should be thinking more about the politics and history of gene therapy and the ethics of genetics, and recognizing both the really powerful positive outcomes and really oppressive outcomes.
Thank you. Yes, please.
Well, I think both of them, the thought that kept on running through my mind is the concept of knowledgeable inquiry creates the precursor for a different kind of dialogue. And it seems like both of you were kind of talking around this issue of this is the way we saw the world before we learned more. And now that we've learned more, we're in that process of trying to find a way to get more people to understand and be involved and engage in dialogue around it, other than just the researchers.
Thank you. Way up here in the back, I think that might be Francois, I can't tell.
I see a clear continuation between both presentations, because a woman who is treated for cancer could end up with a mammectomy, right? An ablation of one of the breasts, or maybe both, and become part of the CRIP community or disabled community.
Thank you.
And that would be a whole new fight.
Thank you. Let's pick someone on this side here. David.
I saw both of you as change agents on the edges. And I think the so what is to change, to recognize, to see what's troubling on the edges, and to change that, which would be good for our students too, and our culture. Thank you.
Thank you. Maybe come back here. Someone had their hand up. Yes, down here, Grace.
I was thinking of healthcare and economic access for both of your lectures. The right to parent. I think there may be fears from expectant mothers about the cost of the child care, and it came up again when you were talking about the environmental concerns. And so there's this concern about economic access. And then again with cancer screening. And you talked about the way that we typically diagnose breast cancer is waiting until there is a large enough lump that it's detected. But by finding these other blood markers, we may be finding more cost-effective and time-effective ways to identify cancer.
Thank you. On this side here, this gentleman right here, I can't quite see who it is in the dark. Oh, I'm sorry, the mic's already there. Okay, good. And I see who that is.
I think there's, from both talks, you get a lot more of the whole process when you observe just not the actors, but like each step, and you divide it up. And once you can analyze the interactions and like the process at each stage and between them, then you get a better view of how everything relates to each other, and how basically you can understand the overall picture better once you get like the micro sections better understood.
Thank you. Bob.
I was thinking of the politics of normality. And especially as I was listening to Maria's talk and thinking about this idea of a normal cell and this idea of an abnormal cell, and thinking of that obviously also when I'm listening to these arguments about ableism and critiquing these presumptions that we all want the same thing. And I wonder what it would look like to even talk to a cancer patient and say, what's the problem?
Sorry, Ben.
It's all right.
So I noticed the tight junctions sort of seem like almost like it's kind of metaphorical for the way that we treat the touchy subjects that Dr. Kafer talked about. The tight junctions the cells aren't able to move to new places unless there's a new entity that comes in, the proteins that you talked about. So I wonder if like the preconceived notions that we have about these touchy subjects, is there some new entity that must come in so that we can break free of like what we are just socially, obviously accepting now?
Thank you. Erica.
Maybe the issue that came up for me was the impact of race, or the dimension of race in Maria's research. The statistics show that Black women are disproportionately affected by breast cancer, get different treatments. So if we talk about the bad future, even with the research, the future will look different for people of different categories, and race is an important one in that.
Thank you. Let's see, let's pop up here to Sebastian.
I saw a clear distinction and I guess focus on the interactions, whether it's between two cells, between two groups that can yield fruitful discussion, and also from a bench to bedside approach. It's really the interaction that we're having with patients and the interaction with people with disabilities that are really, I guess, jump-starting this work and jump-starting these discussions and ideas that we see.
Thank you. Someone over here. Oh, there it is. Yes, please.
I just want to say I saw two almost different sides to the same coin. On the right here, we have a woman who is devoting her life, to cancer research, and I hope that thousands of women may live longer or, happier lives because of you. And to the left, I hope that those with disabilities or injustices may may live fulfilling and happy lives because of your work. And I just look at the two of you, and I see knowledge expressed, through altruism. And I think, that's the beautiful connection here, even though you guys come from two completely different fields.
Great, thank you. Who else do we got? There's someone, way back there. I can barely see up there.
I think kind of what I saw most from it was the emphasis on the things that allow the negative effects to spread. So whether it's the proteins that allow the cells to become more mobile and then cause problems elsewhere, or it's the mechanisms that are used to kind of try to further the divisions between different groups and prevent them from working together. And kind of looking at if you can't really address something arising in the first place, how do you prevent the negative effects from spreading and affecting other areas? So that was what was interesting to me.
Thank you. Yes, over here.
It just seemed that in both presentations there was a thematic undertone of trying to address the root of a problem, whether it be the cause of cancer cell motility or discrimination against people with disabilities. And I was wondering how a more caring ethic or possibly a different ethical framework within our formal political institutions could have an effect upon those different issues.
Thank you. So anyone on this side? Let's see, who else do we got? No one in the center states. The Midwest has not been addressed yet. I've got the coast to coast and then nothing here. Really? Oh, there's an individual right here. Thank you. That looks like Ohio. From where I am, anyway. Maybe Tennessee.
The first thought of being a longtime educator, the first thought that came to my mind after I thought about both your presentations was that this is a very small group that got to hear this today, and if we had the resources to provide more money for research for people like you, but even more important, to get this kind of information out to the public in education, in an educational form, so that we had more people interested in the dialogue.
Thank you. Let's see, who else do we have here? Oh, right here, Michael.
I just continue to be intrigued by what for me was quite surprising, which is the fact that in both of these cases, despite the disparity between the fields, or seeming disparities, we have instances of systems that are conspiring to keep the source of the problem invisible, right? Which I think is actually quite telling and exciting if you think about it. So thank you very much for bringing that to light.
Thank you. Who else? All right, well, while you're thinking, I'll throw out one of mine. So I was inspired by Alison's comment about this phrase, bad futures, which I thought was really interesting, and this idea that both presenters tried to challenge our basic assumption as to what bad futures mean. At the very beginning of Maria's talk, she talked about this abject fear and dread, which was more of an emotional response. And I see those two things actually feeding together in both of these stories, which was surprising for me. So there's one that I'll throw out there. And now, your turn. You don't want to hear more of mine? Yes, great. Whit.
Yeah, to follow up on what you were saying, I was struck by the fact that that idea of a bad future and then self-determination on abortion. I was thinking about Angelina Jolie made a lot of headlines because she had a mastectomy without a cancer diagnosis, but because it was so prevalent in her family, which so she chose to cripple, in some sense, her livelihood, which is a exotic, desirable actress, and striking at her one of her most feminine characteristics.
Thank you. So this idea of alternate insight I found interesting in both of the reflections, and this was inspired by first Alison, actually, when she was talking about how environmental issues in fact are, we know, connected with some of these issues of disability, and that maybe that's the way, in some sense, to fight this, or at least politically to talk about it. And then it got me thinking about Maria's talk, where she's talking about this RNA, this siRNA thing, and how that comes in and just literally slows down the movement of these bad cells. And so you think about, kind of going in and just getting rid of cancer, and these are both alternative approaches, right? To look at the environment as a related but connected idea, or to look at just the idea of slowing down these poisonous cells to not be able to move throughout the body. I thought that was an interesting connection. Okay, don't make me keep going. Yes, this woman right here. Thank you.
Between both of the discussions of Dr. Todd and Dr. Kafer, it just struck me how both address traditional norms of how previous things are addressed, with breast cancer and medicine, and then how institutions address disabilities and injustices among people, and how it just makes people think of other ways, and how actually maybe this is actually the problem, we need to look at it another way.
Thank you. Anything else? Yes, please, Melanie.
Struck me was the titles of the two topics could actually flip.
Wonderful. Thank you. Thank you. Anything else? I can't see. Is there a hand up or someone pointing? Or I can't. No. Okay. Well, I think. Okay. This will be the last one from us, and then I'm going to give each of these amazing speakers 1 moment just to respond to all the stimulation they've heard. Please.
As they were speaking, I was thinking of the TED lectures. And then after this gentleman from Ohio,
Tennessee,
suggested that it's a shame that more people aren't hearing this. I was wondering, can this go viral?
Well, that's a question. You're supposed to be making connections, so you can ask that.
Maybe this can go viral.
That's a statement. Well, I think it's very appropriate to end this first ever event, which, by the way, I just so appreciate the insights that all of you shared that I'd like us to just join in and thank each other for just offering a very unique thing. And then once again to acknowledge and thank all the Brown chair holders and the university scholars, and they're all sitting right around here, and you can thank them afterwards for coming up with this idea and for doing all the hard work in making it possible. It's really exciting when we have individuals from our community come up with creative ideas and then actually take those ideas and do something with it. So I hope this will become a real tradition that we will continue to return to again and again and again. But the final words are going to be from the two of you, and I don't know which one should I ask first. Maybe I'll ask Alison, since she went last, so I'll ask Alison. Just one reflection on what you heard here.
Maria and I have talked about wanting to team teach a class since we first got here. So this whole experience makes me think we should do that. Because it's really interesting how many of you talked about overlaps in language. And so thinking about how language gets used, similar language gets used in different ways in different disciplines, and what the effects of that language is. So let's do it.
Absolutely.
Thank you.
Thank you. I'm very interested in the transition from being a person to a patient, and Alison's talk really resonated with me. I spend a lot of time talking with women with breast cancer, and in one 15-minute consultation with their oncologist, when they find out that they now have breast cancer, they go from being a person to a patient, and they are a patient for the rest of their lives, even if they die of something else and their breast cancer is cured. And I see that in terms of disability studies, that in the blink of an eye, there can be a change in someone's life, and they are forever identified in a different way, and in a way that is so superficial when you think about it. So I'm just in awe of Alison and the work that she does, and yes, we will team teach.
I hope this conversation will continue over refreshments outside. I want to thank all of you for participating and being here. Thank you very much, everybody. Thank you.